And now that we're just about ready, Ike would like to say a few words regarding University House Wallingford and our speaker today. Thank you.
Thank you, Elliot. As a narrow living community in affiliation with the University of Washington Retirement Association, our residents benefit from the partnerships developed with the University of Washington School of Nursing, Social Work, and Pharmacy to deliver innovative programs designed with healthy living in mind. If you're considering making a move to a retirement community, you are welcome to arrange a visit and explore everything we have to offer, including future health options and how our waitlist works. We are pleased today to have with us Dr. Suman Jayadev, who has joined us today to present on a timely topic, an Alzheimer's research update. Dr. Jayadev is a board-certified neurologist at UW Medical Center's Neurological Clinic, director of the UW Neurogenics Clinic, associate director of the University of Washington Alzheimer's Disease Research Center, and leader of its clinical core. She provides services at Harborview's Memory and Brain Wellness Center and the Muscular Dystrophy Association Care Center. She is also a UW Associate Professor of Neurology and an adjunct associate professor of medical genetics. We certainly are honored to have her here with us today. Welcome, Dr. Jayadev.
Thank you very much, Elliot and Ike. I wanted to take the opportunity to welcome everyone and give you all my best wishes. It's been a very challenging past 6 months, and so I'm glad that we could get together today and talk about some of the work that's been going on with on Alzheimer's disease. And of course, despite practicing this several times, it turns out my laptop has several desktops, so I just need to figure out how to share that. So as Ike and Elliot mentioned, I am a neurologist, but also interested in neurodegenerative disease, and our laboratories and my colleagues work on understanding the mechanisms behind Alzheimer's disease. And so today what I'm going to do is talk a little bit about what has been going on at the UW, as well as what has been going on here at the in the community at large. Okay, so now can you see my screen here? Okay. And also, it looked like there was a couple questions in the chat that suggest that the audio wasn't clear. So if anyone's having troubles, please make sure you chat and let Elliot and Ike know, and then we can, and I can make sure that I speak louder, or there's some, we can figure out the clarity issue. So, So we know that Alzheimer's disease is very prevalent right now. There are about 5 to 6 million people in this country alone that have Alzheimer's disease. And because of many reasons, including the the rapidly changing demographic of this country and around the world, we expect to have many more millions with Alzheimer's disease in the coming years. And by 2050, we expect that number to be more like 15 million. And that has significant effects, not only for our personal lives and our families, but also just in terms of the cost to the US in terms of medical dollars. And you know, this is a finite amount of money, so it's important not only from a healthcare perspective, but also from sort of a US solvency perspective to get a handle on the costs associated with Alzheimer's disease and other dementias. And this is a graphic here that sort of goes over the numbers of people that will develop Alzheimer's disease. And like I said, about 15 million by 2050. And that number in cost translates from about 260 billion right now to over $1 trillion by 2050. So this is a significant burden across the board. Just to get everyone on the same page, dementia itself just means changes, worsening cognition. So changes in thinking that also gets worse over time. And there are different forms of dementia. Alzheimer's disease is the most common one. And it has certain features that in general can distinguish it from other types of dementias. So it generally will start off with decreases in memory, short-term memory, and then difficulty remembering names, eventually difficulty with finding the right word. Then there can be apathy, which means not so much depression, but just loss of gumption to do anything. There can eventually be confusion. There can be difficulty with getting complete sentences out to the point where people are no longer able to speak. And then there can be agitation, behavioral problems, paranoia, delusions, withdrawal. Now, there is some overlap in all these features and other types of dementia, but again, these are some of the more typical features of AD. And so this is just a brain image down here below, right here. Can you folks see my arrow when I move it around? Okay, so here is a, quote, normal looking brain. You can see the ridges of the brain, the cortex here. This is the skull. You're looking at a person from the back of the head. And then here's someone with Alzheimer's disease. You can see the relative loss of brain tissue on this side compared to this side. So that's the sort of bigger changes that happen in Alzheimer's disease. And as you can imagine, that does not tell us about what's going on. So we have to go further. So when we define Alzheimer's disease, it's still 100+ years since it was first diagnosed or described. Alzheimer's disease remains, for the most part, a pathological diagnosis. And that's because, like I said, the dementia itself can have a lot of different features in common with other diseases. So it's not until you look at brain tissue and you see certain findings that you say, ah, this was Alzheimer's disease. And these are the findings. These were noted back by Dr. Alzheimer in 1907, and they remain today the features that we look for in Alzheimer's disease to know whether or not this person had it or not. So here is a microscopic view of someone's brain. This purple image here is a neuron. And then this region here is a mix of all the processes, the connections between neurons as they come together. And that's where the information communicates between these processes. So this is outside the cell. In Alzheimer's disease, there are two major things that happen that we know of, and there are many more, but these are the ones we know of now, where inside the neurons, there develops tangles of protein that we call neurofibrillary tangles, and they're made of a protein called tau. And so this tau builds up within the neuron. And then outside the neuron are what are called amyloid plaques, which you likely have heard about. And these plaques are, again, composed of different pieces of protein, but for the most part composed of a protein called amyloid beta. And it's this plaque that has been the subject of a lot of attention, and it's removing this plaque that has been the goal of a number of clinical trials that have been happening over the past couple decades. So in the last 30 years, we've learned a lot of important information through a variety of mechanisms. So number one, like I said, that there is a progressive atrophy, progressive shrinkage of the brain, but that isn't enough to give us any idea about how the brain is shrinking. So in the 1990s, we were able to identify genes that always cause Alzheimer's disease. So these are very rare cases of familial Alzheimer's disease, but those three genes, when mutated, caused the disease. And so we said, aha, so now at least there's something to hang your hat on. There's some starting point. We know that if we start from here, we might be able to get a handle on what's going on with the biology and where to intervene. And so we started making models of the disease in mice and worms and flies using the genes that we know are mutated. And we've been able to get some more information there. You know, there are always going to be caveats with whatever model you use, and I'm going to talk a little bit as they relate to animals, but it's been very helpful. So we have a better handle on what the amyloid protein looks like. We have a better handle on what expression of the amyloid protein does to these models. And then there have been some initial studies on clinical trials. So like I said, the removing the amyloid peptide has been one of the goals, and so we've been able to get some results from that. If you take a drug and you give it to a mouse with the goal of removing the amyloid peptide, you can do that. We've been successful with that effort. So that's important to know that it's possible. It's a big step then between removing the amyloid plaque from the mouse and knowing whether or not it's helpful. And that's been the murky piece we've found ourselves in the last 10 to 15 years. So, and I imagine some of you, or maybe many of you, were with me a couple years ago when we last spoke. And so what I'm going to do is talk a little bit about what I said before and sort of bring you up to date, or bring us all up to date about what's been happening in the meantime. So we have considered the development of Alzheimer's disease to be sort of different layers. And when we are out there in the public and I'm seeing people in the clinic, or you're seeing your grandmother or your daughter, what we're seeing is the way the disease is manifesting in our behavior and our language. But we know that the changes that cause the disease have been happening for decades before. And so here is a graphic that tries to show that. On the right side, so this is a graph where on the x-axis is time, and on the y-axis is change. So low here is more normal, high here is more abnormal, regardless of what we're measuring, okay? And each of these lines are different things that we measure. So if you look at, say, in the green, which is the cognitive ability, we know as you start early, you have normal cognition, but as time goes along, your green line starts to go up and you start to get more demented. Now, what happens prior to this change are all these changes. So changes in the amyloid beta in your CSF fluid, I'll talk more about that. Changes on your brain imaging. So these are, I don't know if you can read this, but these are different imaging modalities to look for deposition of certain proteins. So all these are happening before you can even identify someone as being clinically different. What we found from clinical trials is that the first set of trials where we tried to give a drug that removed amyloid peptide, we found that it didn't really work. But when they did further analysis, there was a suggestion that maybe people that were earlier on in the disease, so not people out here, but people out here, may have had a benefit of the drug, may have had a benefit of removing amyloid peptide. And that, sort of becomes intuitively coherent with what we would think, because what it's telling us is that if you are so demented, or if you are so far along the process that you already are demented, you've already lost neurons, then giving you a drug to remove the irritant may not be that helpful. What we need to do is stop the disease or slow the disease way back in the early phase where things are still changing. So that was one of, I think, some of the big takeaways that we got from these clinical trials. So they may not have been on the outset helpful insofar as we found a drug, but it did give us a lot of important information that's sort of driven the pace of the field in certain arenas. So this is where we find ourselves, that there are challenges and there are now endeavors or goals that we have to get to a therapy. And I think it's given us a chance to kind of regroup and think about how we can incorporate new technologies, new opinions. The NIH said a couple of years ago that their goal was to get a therapy for Alzheimer's disease by 2025. That's 5 years from now. I think the reality is that given some of the changes over the past year in terms of priorities and need, that may not be the case. The NIH and other scientific efforts have had to be redirected towards COVID. But nevertheless, I think it reflects this big, huge Herculean effort they're trying to mobilize. And so what they've done is they've massively increased funding to Alzheimer's disease. They've also done this thing where they've reached out to people who are not typically in the field to say, hey, you work on cancer or you work on cardiovascular disease. How would you think about solving this problem in Alzheimer's disease? And so that I think has been super helpful because it gives people, it's a fresh pair of eyes, et cetera. It's also a fresh pair of tools. So I have a lot of reason that I think we're really on the right track. So these are some of the challenges we already knew about, and we probably as a group have discussed before, that when we study neurodegeneration, Ike and Elliot, do I have until 11:30 or 12?
11:30 is what point we'll start taking questions.
Okay. So I hope everyone's writing down their question if I skip over something. But the challenges are that we can't take a piece of your living tissue when you're still 50 so that I can understand what's happening early in life. The animal studies don't always properly recapitulate the processes of dementia. You know, you can't really make a demented mouse. You can make a mouse that has movement problems, but you really can't accurately find out if a mouse is having early language problems or can't find his keys more often than not. That there's a big genetic diversity, not only between humans and animals, but also between all of us. So not just, you know, and we know that already, right? So I got a bit more color than Elliot does. And there's a lot of other changes that we don't even see that are, you know, inside the brain, the way our immune system works, the way our response to a little bit of injury works. Those are differences among humans that occur over a spectrum, and all of that will impact the way our Alzheimer's disease develops and also impact our chances of even developing it, Alzheimer's disease. And so that's all, you know, what is really encoded in the genes. And also, I should say, what's also encoded or the reflection of the way we interact with our environment. So that's this gene-environment interaction. And then the last piece is what we just mentioned, which is that by the time we identify patients with Alzheimer's disease, it may already be too advanced. The horse is already out of the barn. So what we need to do is develop early biomarkers. We need to identify the Alzheimer's disease mechanisms that are relevant to the person coming into the office. Now, that's a very simplistic way of saying it. I'm not going to say that we have personalized medicine for Alzheimer's disease. You know, we're not going to have, how many of us are here? You know, there's not going to be 40 different AD therapies, but maybe between the 40 of us, there may be 6 of us that have this type of Alzheimer's, 10 of us that have that type of Alzheimer's. So can we try to start to separate out what's going on with different people? And then we need to think, and we have been thinking about different models beyond the animals that we have to really look at Alzheimer's disease. And so the innovative models really comes in with new stem cell technology that I will be talking about, assays or ways to look at individual risk or look at each person's Alzheimer's disease, and then new imaging technology and other biomarkers that can be gotten out of the blood and the CSF have been developed recently, which will also be helpful to identify people early. So Alzheimer's disease leads to neuron death, and it's the neurons in which the information is encoded. It's the way the neurons talk to each other that memories get laid down, so that, you know, they're key. However, all the other pieces of the brain are critically important to keeping neurons alive, to making sure they connect the right way, to making sure they develop properly. And so when we think about Alzheimer's disease now, we really need to think about what are all the different cell types that are going wrong, in part because it's important to understand the biology, but also because it may be a more tractable way to treat the disease. If I can't rescue your neurons because they're just too complicated of a cell, maybe I can at least protect them by changing the way some of the cells around them are behaving. So this is a complex graph, but, you know, each cell type is a different color. So here you have I'm just going to go to this side. So this is the disease side. This is the normal side. So here's an astrocyte cell. This cell interacts with the blood vessels in the brain and then communicates with the neurons that are in the brain here. These microglia, these light blue ones, communicate with the neurons as well as the neuron processes. So it's all a big symphony of cells. Now, so you can surmise then what I'm getting at is that disease from Alzheimer's disease isn't just because you have amyloid beta injuring neurons, that there's a bunch of different things going on. Now, we found more direct evidence of that, or more suggestive evidence of that, when we started to look at genetic studies of people. So I told you early on that there were familial forms of Alzheimer's disease, and from there we identified 3 genes that always cause AD. And so we said, aha, the way they work is probably influencing the way Alzheimer's disease develops, right? But those people are barely a couple % of the population that have Alzheimer's disease. So what about the rest of us? do we have any kind of genetic risk factor for Alzheimer's disease? And then will that tell us about the biology of what's going on in us? And so here is a graphic that tells us the information we have about that question now. So on the x-axis here is the frequency. So each of these circles is a gene change, okay? I'm going to call it a mutation just because it just means gene change. It doesn't mean anything really more horrendous than that. So each of these circles are gene changes. The words in here are each gene. So there's ApoE4. This is one version of ApoE. TREM2 is another gene. And these are other genes down here. Okay, so each of these circles are genes. This x-axis is the frequency with which there is a particular gene mutation. So this, these genes here, these gene mutations happen a lot. 50% of the people have gene changes here. So they're not really necessarily causing disease on their own, because 50% of the people don't have Alzheimer's disease, but a lot of people have these little changes. Very, very few people have these changes. Like these are the ones that always cause familial Alzheimer's disease. Now on the y-axis is the risk of getting Alzheimer's disease if you have this gene change. So right here, these are the early ones I was talking about, where if you get the disease, if you have the gene change, you have the disease, so they're super high. These ones down here that I talked about, where a lot of people have them, as you can see, if you go all the way over to the y-axis, very low risk. So the point is that we all have many of these, okay? So if I look at myself, I probably have 27 of these, but they're all super, super low. It's the combination of having all those gene changes that in the end, in aggregate, changes my risk for Alzheimer's disease. That's point number one. Point number two, each of these genes are actually related to different pathways in the brain. So they are run by different cells in the brain. These are not all neurons that have these pathways, and there are different pathways. So APP metabolism, that's the amyloid beta story that we know. But then there are things like the immune response. So gene changes in the immune cells can give you a higher risk for Alzheimer's disease. Gene changes in the endocytosis. Endocytosis is the way you traffic protein within a cell. If you can't traffic your protein in and out of the cell properly, your cell has dysfunction. So different ways to get to Alzheimer's disease, run by different cells. I just put this in here because I know that that was one of the lines in the introduction, but basically what this means is that we can start to get a more precision approach to Alzheimer's disease. So here on the left is what we've been doing. You have a bunch of different people. They all have different layers of genetic risk. They all have different exposure to different environments, so they're all different colors. And then when people start to become symptomatic, they get one pill. The approach that we're hoping to do now is to take all these different colored people and then start to divvy them up by their genetic risk, like I just told you. So let's say I'm going to find everyone that has a lot of these blue circles because they're immune related. I'm going to put them in one group. Then I'm going to find all the people that have a lot of this cholesterol stuff, and I'm going to put them in another group, okay? So I'm going to divvy them up and say everyone, and then everyone who smokes is going to be a subgroup here. And then I'm going to give them the right drug. So that's the idea of the precision medicine. It's not necessarily the right, one person, but it's to give people the right drug that's going to address the type of Alzheimer's disease. The importance of biomarkers. So this is an example of how in living people, a biomarker can be quite helpful. So this is a scan of a brain that will identify amyloid beta. So red means there is amyloid beta. When there's no red, that means there's no amyloid beta. This is a normal person that doesn't have any amyloid beta. That's consistent with not having Alzheimer's disease pathology. Here's someone that has Alzheimer's disease, and they have a lot of amyloid beta pathology, and that's consistent. Here's someone who has early Alzheimer's disease, mild cognitive impairment, and you can see they already have a lot of amyloid beta, right? They've already deposited a lot of amyloid beta. Now here's someone who looks on the outside very similar. Both these people have mild cognitive impairment, but this guy doesn't have any amyloid beta. So if you give this person who was diagnosed with mild cognitive impairment the same drug to treat his dementia that's actually targeted for someone like him, it's not going to work. So that's one of the values of biomarkers, is to make sure you're actually detecting the right disease in a living person. Sorry, when I take the old slides, I also get my son's nonsense here between the different slides. There is also efforts to do different types of imaging, such as what's called functional connectivity. And this has been a really interesting way that people have looked at all sorts of things. Like neuroeconomics is actually a field where they try and see, you know, how you respond to certain advertisements. But basically, here in this image, when things are lighting up, the parts of the brain are active, and you can see which parts of the brain are acting in concert and which ones are not. And what we know is that normally there are things that are acting in concert, but with Alzheimer's disease, they start to lose their connectivity. And you can see that image here. You know, these two are not as connected as before. And so you can you can use this information also to identify people earlier. And then there began to be the ability to get biomarker information from blood. Let me take a step back, because probably when I spoke last, or the last 5-10 years, or anyone who's been to the Alzheimer's Center has known that we generally ask people for a CSF sample. So that's the fluid that goes around the brain and the spinal cord, and it's a really useful substance because it's direct contact with the brain. So any type of degraded protein or any problems or immune problems, you can measure that in the CSF. The problem is you don't want to have to give someone a lumbar puncture every time they come into your clinic to see whether or not they've developed early pathology with Alzheimer's disease. So there's been a lot of effort to get blood-based biomarkers. And so a couple of years ago, there was studies or groups that were able to detect amyloid beta in the blood. And so that was helpful, because otherwise, like I was telling you, you had to wait until someone was dead to find the amyloid beta in the brain. But then we were able to develop imaging where we could put someone in a scanner and find out if they have amyloid beta, like those images I just showed you. But now you can even just draw it from the blood. This is all preliminary. We're not doing this in the clinic. There's no data yet to confirm how predictive it is, but this is just the way the field is going. And so there has now been a growing array of these fluid biomarkers, and they all are sort of related to the cells that are in the brain. So in the CSF, so again, that's that fluid around the brain, you can measure a number of things. One of them is the tau protein, and tau is that one of two proteins that we saw in the autopsy that's in the neurofibrillary tangle in the brain. So A beta and tau, this is tau. You can see that released into the CSF. You can measure that. You can also measure the tau in the blood. You can measure evidence of inflammatory markers that are released from the astrocytes in the brain and the microglia in the brain. These two cells have immune function, so you can measure the degree of inflammatory change within the CSF and to some degree from the blood. And then there are newer imaging modalities that are actually targeting aspects of neuronal connectivity. So there are now protein or molecules that are targeting the axon terminals, the connections between the neurons, a compound that will bind there so you can see the integrity of the connections between the neurons. So that's super exciting. This is all really still on a research basis. No one is using it diagnostically, but the hope is that you can use this in the clinic, but also in the clinic as in the sense of detecting people really early, but also can you follow these over time and say, is my drug working or not? There was some discussion, and I'll get to that. So here's just an imaging picture of some of these other modalities. And so this is again, the person from the back of the head, okay? And red is a high signal, green is a low signal. And what you see here is if you can use a compound that identifies amyloid beta in an Alzheimer's disease brain, you see that there's a lot of amyloid beta deposited here, and it gives you a pretty nice idea. In some people, and it also can give you a general idea about what parts of the brain are affected. You can see that more easily here. This is a scan that identifies the tau protein, and you can see, oh, okay, gosh, this person actually has a lot of tau that's asymmetric, but this certainly is abnormal, and I can do this in a living person. What you can also do is just also look at the metabolic integrity of a brain cell. And so here, the absence of red is telling you that these cells are not functioning and using as much sugar and blood flow as they should. And then the imaging I mentioned earlier, a compound that can actually bind to the synapses being the connections between neurons, and you can look and see, okay, this person has, so this happens to be a control person. You can say, okay, well, we have a normal looking pattern of synaptic health. And the argument here would be there's reason to believe that the loss of this synaptic integrity may be happening earlier than some of these other overt patterns that we find here. Because really, in order for us to find a tau protein on a scan like this, there has to be a lot of it. Okay, and so now we have biomarkers that that can be useful at different stages. So we can have biomarkers that you can pick up in people that are cognitively normal, biomarkers that are still changing as you go from mildly cognitively impaired to dementia. And this is really great to sort of fill in the gaps, because, you know, the other thing too is we don't want to just target Alzheimer's disease therapy to people that aren't demented. We really think, you know, there's still room to go for us to be able to treat people that have already gotten a diagnosis or already have mildly cognitive, mildly impaired status that we can at least slow the progression for. There was some excitement over the summer about another blood test. It's really along the lines of what we just talked about, and it is to identify that tau protein, which is, again, the protein that's in the tangles in the brain. I told you that there was that blood for the amyloid, so now this is blood for tau, and we can detect it in blood. And there are different ways that that you can measure tau. That's what this graphic is. You can measure tau in the CSF, and you can measure it in the blood. And so what these two studies showed, I didn't pull them up, is number one, you can use tau to distinguish between different types of dementia in their study. So that was useful. Someone comes in, they have some behavioral changes, and they're a little bit younger than you'd expect, say they're 65 or something. You're not really sure if it's Alzheimer's disease or a different form of dementia called frontotemporal dementia. This blood test can give you information that might be able to help distinguish the two. The other thing another study did was to demonstrate that they were able to detect the tau protein in the blood before people were symptomatic. So that's pretty important too. And these are all studies that need to be reproduced. So that's a little bit of information about biomarkers. And then the other stuff, I want to talk to you about though is like, how are we going to get at what is going on in the brain? Because we really need to start putting emphasis on understanding the biology of the brain in order to create the therapies. There is so much interest and so much enthusiasm and heart that's going into developing Alzheimer's therapies that we want to make sure we're not cutting any corners. We want to make sure that we really understand the biology, because, you know, it takes us a lot longer than some other fields to get to where we need to go because of those problems I told you about. You know, we can't get fresh tissue from a healthy brain. We're limited a lot of times to autopsy tissue. So this is where, you know, this is where we are. We come across basically this bicycle here. It's broken, it's rusty. You don't know what happened, but this is what it looks like. And so our goal is to figure out, how did it get here? So, you know, we know there's this Platonic idea of what a bike should look like. We know somehow it went from here to here. Some of the tools that we've been using are the following. One are induced pluripotent stem cells. And so those are stem cells that you can create to form any type of brain cell you want. And so we can, in the dish, start to create the pieces of the bike. We can create the wheels, we can create the gears and the seats, manipulate those, try to see what makes them work, try to see how they fit together in order to make a functioning bike, and then try to abuse these pieces and see what looks like, you know, what kind of injuries make these clean little pieces look like the ones that we find in this broken, rusty brain. The other way is, and this is sort of what the animal models do, is you can take this beautiful bike and just throw it down a ravine and see, okay, what kind of big problems can you do to this intact bicycle to break it up? So those pieces of the bicycle, these are some of them here. So there is the neuron. the astrocyte, the microglia. I showed you a little bit about that in some of the earlier slides. And there are other cell types too. There's something called an oligodendrocyte, and those cells are very important for the development of multiple sclerosis. They're also important for the brain as well. But so when we think about trying to study Alzheimer's in the dish, we have a lot of emphasis on understanding the neurons in the dish. And in the dish, I mean, you know, at the lab bench. But there is interest in studying the other cells as well. And so my lab happens to work on inflammatory cells, and there's reason for us to think that. There's, well, like I showed you, risk in inflammatory genes can increase, or mutations in inflammatory genes can increase your risk for Alzheimer's disease. So we know that they are relevant to developing the disease. They're not just reacting to what's happening. And we know that increased inflammation happens in Alzheimer's disease brain. So here is another one of those images like I showed you. But this time it's not, this scan is not labeling amyloid beta, it's labeling activated microglia. Microglia are the innate immune cells that are in the brain. So once the cell becomes activated, it picks up this molecule, and you can see there's just tons of inflammatory cells in the brain. So one of our goals is to say, you know, we got a team that's working on trying to protect neurons. We need another team that's going to try and figure out how do you quench the inflammatory response, and let's see if we can all get these to work together. I think I just told you this, microglia, these guys are important for responding to the plaque and the tangle, and they're important for clearing the plaque. So microglia, what they do is they get rid of debris. And what you want to do is get rid of that amyloid plaque. One of the medication goals was to use a therapy, a drug to get rid of the plaque, but your brain is also doing it on its own, using its own immune cells to get rid of it. Okay, so this is the disease in a dish model. You start with a human, and you take a piece of his skin or a piece of his brain, the tissue that surrounds the brain, and then you reprogram those cells into becoming stem cells. And what they can do once they're stem cells is turn into any brain cell you want. So now what you have is a dish in which you've got brain cells from someone who had Alzheimer's disease, and how his cells function. So not some cell line you got out of the freezer, not a mouse cell, but a human cell that has a human genome, and a human genome with risk for Alzheimer's disease. And if you plate these, you put these cells in a dish, you can actually put many of them into many different wells of a plate, and then you can screen compounds, you know, from some pharma company, many at a time. This is just an image of what they look like. You start with skin cells, you make them into stem cells, and then you can create these immune cells, these microglia, or you can create these neurons, and a bunch of different cells. So you do these all in the dish. So here is an example of why doing different cell types is important. And it's just an example of what we can do to get a better understanding of the mechanism behind Alzheimer's disease. So one of the genes that was on that little table is called SORL1, and I misspelled it here. SORL1. It's a risk gene. So there is a fair number of people that have some little change in their genome that may affect the way the SORL gene works. But SORL is very important for the way the cell moves proteins through the cell. So here is an example of what it does, okay? This is the inside of the cell. This is the outside of the cell. Here's an APP molecule. Now, APP is the protein that when it gets cleaved, it releases this little piece of its protein called amyloid beta, and that's the amyloid we've been talking about all the time. So when APP gets cleaved, it releases the amyloid, and then when this gets out of the cell, it gets deposited everywhere, it causes mayhem. SORLA, this protein that we're talking about, is responsible for helping to chaperone APP to the right place. So when it's working, it takes the APP and it moves it somewhere else, and then APP moves along its way. When SORLA is not working, this APP gets stuck in the endosome. It gets stuck here, and when it gets stuck here, it just starts getting cleaved. So you get more and more of this A beta. So instead of moving the APP away, it gets stuck, and then you get more and more and more A beta. So you can imagine why loss of sorrel function could be problematic. The problem is, what cell type is that doing? So what Jessica Young, our colleague at the UW, did was to remove that gene from her neurons, from the stem cells, and then find out what happened. So she created a stem cell line that was absent in sorrel1 called the knockout. And what she first did was just look to see what did these endosomes look like. And what I didn't show you here is a normal cell, but here's this beautiful neuron in red. The EEA1 is a green marker for the endosome. And what you see on the close-up here is that they should all look like this, but they're little ones. So when they quantified it, the absence of sorrel1 changed the size of these pieces of the cell that are responsible for trafficking protein. When we made microglia, when we made the immune cell from the same person, we didn't see it. So this was how we said, we know there's a risk gene for Alzheimer's disease. We don't know what it's doing, but if we're going to create a drug, we have to know which cell type to target, because we have to know which cell type is doing something that's relevant to Alzheimer's disease. And in this data, it would suggest that right now, the sorrel gene is probably more important in neurons. So we did even further studies, and we found out, the point of these graphs are really that if you take out sorrel, okay, this is what this knockout is, you don't internalize the amyloid beta as well. So your cell can't get rid of amyloid as well. That's only in the neurons. When you do it with the microglia, the immune cells, there's no effect. So the take home from all these graphs are that the sorrel gene is more important in the way neurons interact with amyloid beta rather than microglia. So that's super important, because trying to improve the microglia's ability to clear amyloid beta may not be the relevant pathway here. It's got to be targeted to the neurons. Okay, this is just a reminder that right now we've been talking about cells in the dish. One cell type here, another cell type here, but really, right, there are tons of different cell types, but there's also a specificity in the way the brain cells work. So, you know, the front part of the brain is more executive function. This temporal lobe part of the brain is more memory. So where does that specificity come from? Why do those function differently? There are different parts, there are different types of neurons and different types of cells even within those brain regions. And really it's the communication between all of them that are really important. And when the brain is developing, when you go from just a 5-cell blob into this complex brain, it's because each of these cell types are communicating with each other in order to mature and grow properly. This is just a very messy graphic, just to give you an idea. I mean, each of these little circles are packets of communication. All of these cell types are communicating with each other constantly. So on one hand, it's interesting to take just neurons in the dish and see what happens if we perturb something. On the other hand, we're probably missing a lot of input from the other cell types. So how do we reproduce that? And that's where these organoids come into play. You may have heard them, it's called brain in the dish. And basically, it's similar to the way we make stem cells. You grow them in the dish, but then you allow them to create a 3D structure. And amazingly, they organize on their own. So within their genome, they've got the information to tell each other where they have to layer up and where they have to travel. I mean, to some degree, that this is, it only goes so far. So here's an example of a 20-day organoid. You can kind of start to see a little bit of this organization here. And this is from Jessica Young's lab. And then if you take a section of this organoid, you can see how incredible it is. It organizes by itself. We started with just one type of cell, put it in the dish, and then it did this, with some nudging. But so here what you can see is that in this 3-month-old organoid, it just sits in the dish for 3 months, that it starts to develop early neurons, these red ones, early developmental neurons, and then you have more mature neurons in the green starting to grow. And these guys essentially recapitulate the timing of a growing fetal brain. So this is, you know, sort of similar to a 3-month-old fetal brain, maybe a little bit older. But this way, at least, we can start to look at what does genetic risk, or what does the lack of oxygen, if you're trying to mimic, you know, high blood pressure or something, not lack of oxygen, but poor blood flow, what does that do to the way the cells talk to each other, not just alone? Okay, so you know, there have been a lot of technologies that have been developing over the last couple of decades. And the neat thing is that there's been a lot of effort to try and merge all these. And so there, the NIH had started something a number of years ago called AMP-AD, which was meant to be a consortia across at least 4 or 5 different groups so that they can kind of merge data. There's another endeavor called Treat AD, and the UW is part of that, where it's a mix between computational biologists, neuropathologists, cell biologists like us at the UW, neurologists, medicinal chemists that all they do is just make drug, and then organic chemists. So it's really a multidisciplinary approach to just sort of start from scratch and see what we can do and really make this an informed decision that is drawing directly from patient tissue. So this is sort of what we've been doing or has been done. So basically you start with people that are normal controls or Alzheimer's disease. In this case, and in this study, what we're doing is starting with patient brains. And then you analyze them across all of these measures. So you measure their genes, you measure their protein, you measure the way the genes are modified. So you can measure all of that across thousands of people, okay? And that's where the high throughput sequencing has been very helpful. You can do this more cheaply and faster. And so you now create huge amounts of data about all the different genes and all the different cells and thousands of people, but now we have better bioinformatic approaches to try and make some sense out of that, to try and see what is the underlying structure of the Alzheimer's disease brain compared to the control brain in terms of genes, et cetera. So you can reconstruct now biological networks based upon the genes that you measure and start to see what are the genes that seem to come up to the top that are, you know, what are the networks that are particularly abnormal. And so this is a table, a graphic from one of their papers, which is basically these are some of the genes that they said, you know what, these clearly are driving downstream processes in the Alzheimer's disease brain. These are the ones, guys, we should think about how we're going to target. So right now you have this list. So right now the medicinal chemists are trying to understand how do you interfere with this interaction. Our group, Jessica and I, are trying to say, well, what happens if you just get rid of this gene? Is it doing what you hypothesize to do? Or is it something unrelated, you know, before we start spending all this money? So it's a lot of work going on in parallel. So before, when I had talked to you, I think, and I had said, look, we've got stem cell biology. We're doing basic function studies in the dish. We're then going to identify targets, identify the genes and the proteins that are abnormal in these Alzheimer's disease cells so that we can then test compounds. In addition to that, now we are, with our colleagues, they have now produced from bioinformatics candidate genes that we should pay attention to. So we are going into our cells. We're using CRISPR editing to remove those candidate genes, go back and make our cells, and then try to identify pathways, and then inform our colleagues about model systems and fly and zebrafish and other models to test more compounds in a high throughput method. All right. So that was to explain how we would use stem cell biology to reproduce certain brain cells and test function and understand the pathology of biology of AD. The other piece that I was alluding to with that bicycle analogy is that we also have better ways to look at the damage that's occurred in the brain itself. And so, you know, we have microscopic ways. So here's a picture, you know, microscopic image of the brain, but what's really happening, because we know that if you take some of that tissue, so the gene expression I was telling you about, that is when you take a chunk of tissue and you measure the genes. The problem is when we do that, I don't really know if that gene's coming from the neuron or the microglia or the astrocyte. Our bioinformatic friends can make some sense of it because they can say, well, Sumi, this gene is never in any other cell but a neuron, so we know it's got to be a neuron. But it's not often that we're that lucky. So one of the other challenges has been, how do we figure out what the pattern is in the right cell type? So that's where the last couple of years, there's been an effort to do sequencing on each cell. So you can take a cell, you sequence it, and then you say, this cell has high amounts of this marker, low amounts of that marker. It's not perfect because you don't actually get all the genes. You just get some of the genes. But because you have thousands of genes, you can hope that you get a reasonable picture of what's going on that particular cell type. So this is just a graphic of how we do it. But the point is, we're going to get gene expression data for each of these type of cell types, okay? So just to give you an idea of what the scope is, because I think this is really important, there is a lot of great technology out there, and there's a lot of groups that are really excited about implementing that technology, and we always have to be aware of where the limits are before we can start extrapolating from there, and extrapolating what we can do with it. So here is probably the biggest study that's been published so far. It's 48 different people, and from there they collected 80,000 cells. It's not a lot of cells per person, okay? But what they were able to show using this approach, and this was really interesting, is that if you start with just people that have early disease, early Alzheimer's disease, you can find out where you can see genes that are changing in particular cell types. And so on the x-axis here, these are names for different cell types, and the degree of blueness is the amount of change in the gene. And you can see that, you know, not all these genes are changing in all cell types. It's really only in some cell types. And so knowing that these genes are changing only in one cell type or the other is allowing us to kind of put the mosaic together and figure out where the pathways are changing early in the disease. If you do that same analysis in a late Alzheimer's disease brain, then you see that really there's little change, right? Because by now everyone's just sort of on their way out. All these cells are just gasping for their last breath. So it's important to have a sense of what stage of disease we're looking at. Let's see, I think this is really interesting. What you can then do is try to understand, so you have gene expression from different cells. What you can do then is put side by side those cells in terms of how similar they are. And you can see how a cell evolves from normal all the way to a disease state. It's like, you know, you took the fish that came out of the ocean and you watched how it went from fish to mammal to primate to human. Same sort of thing. But instead of beings, we're going to use a cell, and we're going to use a cell that we got from different types of people. So what you have here, each of these circles is a cell, and they are collected from people that are either controls, so have no disease, or Alzheimer's disease, let's say here in red. And what you can see is that in general, these neurons are kind of similar over here when they're controls, but they eventually start to change and eventually start to become diseased. And the same way with the microglia. I don't know how well it's coming up here, but you can start with normal microglia and then they start to become more and more diseased as they start to become red. But what you see here in the microglia is that there are different groups of red. So there are different types of disease, Alzheimer's disease microglia. which is important because it turns out there's a lot of different types of microglia. So then you can imagine our question is, what are they doing? How do we interfere? And that's really, and then that's what some of that bioinformatics does. And so here's a study that just came out. This is what they did. They did what they took the microglia from the brain, they isolated it, and then they just put them together on a line and tried to figure out where they divided up and how they progressed. And what they did is they started, so here are normal microglia from a normal brain. And you can see that they start to evolve, but they can evolve this way, whoops, or they can evolve this way. And if they looked at the profile, the gene expression of these different groups, they saw they were quite different. Some of these are the type of genes that you'd need if you're trying to get rid of amyloid. Some of these over here are the genes you need in order to move through the brain. And these genes, these cells are just burnt out. They're halfway dead. So now we can say, okay, well, what are the gene changes that happen when you go from normal to moving or normal to responding to amyloid? Can I turn on that gene artificially then? Can I try and make my microglia be more responsive to amyloid if I can kind of replicate the changes that are happening here? It's sort of a simplistic way of explaining it, but that is the general idea of what you can learn from watching the way cells normally change in a disease course, and then try and leverage that information to create better drugs. So at the UW and in collaboration with SAGE and now, like I told you, our friends at Emory and Stanford and a bunch of other places with that Treat AD, all these different groups are coming together to figure out the way cells work, which person's got what type of AD, who's going to develop early, and then try and make a better informed therapy with multiple prongs to it, because it's not going to be simple. It's not going to be a smoking gun, or not a smoking, it's not going to be a silver bullet. It's going to be, a cocktail of drugs. So I think, let's see, did I do it? I did it. And I'm open to all sorts of questions, because I know we probably sped through some things, but happy to talk more.
All right, thank you, Dr. Jayadev. We really appreciate your time today and the true wealth of information that you provided. We've gotten 3 questions so far, so we'll get to them in the order they came in. While we are getting to these questions, feel free to ask any more in the chat feature, or again, if you're joining us by phone, you can email them to me at elliot.hack, ELLIOT, .HAACK@ERALiving. And please let us know if you're having any issues with the audio. I understand that there was some audio distortion earlier, so if you're experiencing any of that, please let us know in the chat as well. The first question, Dr. Jayadev, that came through asked if there are any studies currently in Seattle that need subjects, and if so, how can one get into a study?
Yeah, that's a great question. And there are different types of studies. So there are, there's going to be clinical trials for drug that, and I'll, this is the good thing about Zoom is I can pull up the website. Then you can always look at that and see if they're interested in someone like you. So, whether if you're control or not control, or male or female, et cetera. Then there are other research studies that are like, so the Alzheimer's Disease Research Center, what that is a longitudinal study, and that's what I'm a part of, where we see people once a year, we collect blood for the purpose of those biomarkers, and we just follow you year after year. And then if there are other research studies coming up, then we say, hey, are you interested in doing that? So for that study right now, what we're recruiting are people that have some mild cognitive impairment, or people that don't have mild cognitive impairment or cognitively normal, but are willing to undergo a lumbar puncture. So if that is you, you can let us know, and I'll, and I think Elliot has my contact and he'll give it to everybody. I can tell you more about that. Everything is sort of on hold because of COVID. And as a side note, it's really interesting because the scientists had to put things on hold. We had to shut down labs and all that, but at the same time, it was really difficult to figure out how are we going to keep seeing our people once a year for our clinical studies. And so in the meantime, in fact, one of the docs that I work with for the Treat AD, he's also the chair of the clinical core studies with the Alzheimer's centers, and they've retooled their studies so that we can examine people over the telephone and over Zoom instead of having to do them in person. Anyway, the short story of that being that things are a little bit slow right now to recruit new people into the Alzheimer's Center, but that is another way to get involved. But what I would say, and this is hopefully what you can send, Elliot, is that there at the Memory Brain Wellness site is a research tab, and you can see all the different studies that are going on in the region.
Thank you, Dr. Jayadev, and we will share Dr. Jayadev's email address with all the attendees afterwards, so you will be able to reach out to her directly regarding those studies. The second question that came in asked, has there been research that looks at nutrient deficiencies as a precursor to developing Alzheimer's disease?
So that's a very good question, and it's really important because it's preventable and it would be very useful to know. I don't know offhand that there's been anything that's certainly proven. There's been a lot of effort in terms of the benefits of exercise, but I don't necessarily know about a specific nutrition deficit.
Okay, thank you. And then the third question that we have, and remember you can still submit questions in our chat if you have any that you'd like to ask. But the third question is, can one have clinical symptoms that could be a harbinger of dementia, but that are caused by something else, such as fatigue, depression, illness, or something else?
Absolutely. Absolutely. And so, and I can just tell you just from my experience when we sit, so with this study, this Alzheimer's study, we have a consensus meeting. So every year people come in, they get their tests. The neuropsychometrists do their thing, and then we meet once a week to go over the week's evaluations. And we have many times seen some in where we've said, this seems like mild cognitive impairment. And then the next year, they're fine. So yes, you can even get to the point where you were even deemed by an evaluation to be impaired and yet come back later and be okay. So there are a number of things. So just in terms of the typical day-to-day fluctuations, sleep, blah, 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 that can impact the way you think. But depression clearly can mimic dementia. In fact, it's even called pseudo-dementia. And it affects your attention. It affects the way you can bring information in and even keep it there. So sometimes neuropsychological testing can be helpful to distinguish those. But certainly having a discussion about depression in general is important. Sleep is very important. So sleep apnea can mimic can mimic dementia easily. And that's another one we've seen where people seem like they're getting demented. They have very poor memory. They can't attend. They don't remember names, but they start on their CPAP machine and they get much better. So that means going to a sleep specialist, finding out if you have sleep apnea. What were the other questions? But yes, so there are systemic things that can mimic dementia that are worth talking about and thinking about. And the other piece too of it is that we all lose the keys. We all forget someone's name, et cetera. So it's unnerving to know what's real and what's not, or maybe more nuanced, what's just typical aging and what's not. And so that's understandable. It's an understandable worry, but really that eventually makes itself clear. But those are the types of conversations you can have with your doc. And there's been a lot more effort made now for the dementia docs to talk to, or to talk to the primary care docs to say, these are kind of things that you can screen for, to talk to your patients about.
Thank you. We had two more questions that have come in. The first one asks if there have been any studies on how the environment affects cell and brain processes.
Yeah, there are a lot. And I have a colleague, Gail Lee, who's particularly interested in the way air pollution can influence the way the brain works. So there's a lot of reason to think that. Animal models are also used to do things like manipulate their environment, and they can, show worsening of whatever the disease model is. So there are definitely effects. I can't tell you specifically about which ones are most relevant, But like I said, pollution is certainly one that's of interest to people.
Thank you. The next question was, how prevalent is heredity in Alzheimer's disease? If there's no Alzheimer's disease in your family history, what is the risk?
Fantastic question. And recall that I am a neurogeneticist, so in my mind, everything is genetic. But so the rare forms of Alzheimer's disease I was talking about, where if you have a gene mutation, you will develop Alzheimer's disease, is exceedingly rare, less than 1%. Now, what we do know though, is is maybe 75% or 50% of people. So if I just look at Alzheimer's disease though, it's more like 25% of people have some other family member with Alzheimer's disease. And I think that number is actually higher. Those are older statistics. So, and we know that there is some sort of genetic contribution. Now, the absence of Alzheimer's disease in your family, I can't tell you for sure how decreased your risk is. What I know is that if you have a first degree relative, you have maybe a 10, 15% increase higher risk. So, there is still a risk of Alzheimer's disease. It increases with age, and there are other factors that may not come up with the family history that are still going to be influential to developing Alzheimer's disease.
All right, thank you, Dr. Jayadev. If we have no further questions, then I think we're just about ready to wrap up. Remember, we will be emailing out a recording of the webinar today to all of you, along with the slideshow, so you can peruse those at your convenience, as well as Dr. Jayadev's contact information if you have any questions you'd like to direct her directly to her. We will also include a survey sheet that we'd like you to fill out and tell us about your experience on the Zoom event today. We're always looking forward to hearing from you. We're interested in hearing any comments that you have so that we can make this experience even better in the future. We will look into what those the audio issues may have been at the beginning, as many of you have joined us before, so you know we haven't had that before. So we'll look into it. Hopefully it won't come up again. And then I'll turn it over to Ike to say goodbye.
Okay. On behalf of University House Wallingford, thank you all for joining us today, where life goes on and lifelong learning thrives. We appreciate your attendance, and thank you, Dr. Jayadev, for such an informative presentation today. Thank you.
Thank you all.
Have a good day, everyone.
Thank you.